Okay, so this kind of just gives an overview of like the history of DNA to what we know today. Okay, so just to kind of recap on what each scientist ended up figuring out. So the central dogma is that there's a flow of genetic information in a cell. We go from DNA, it gets replicated, then it goes to RNA through transcription, okay, and then we go to a protein through the process of translation. So we're going to start with replication, okay? So always remember replication happens during S phase in interphase, okay? So copying the DNA, you have to have a template strand. Okay, so that template strand is the thing that we are going to use as kind of the base for replicating. It is a semi-conservative process. It happens during the S phase of the cell cycle, and there's a Hayflick limit. I'll go over what all of that means with the telomeres. Okay, so DNA replication. Basically what it is a whole bunch of enzymes that coordinate replication at multiple locations at the same time. It kind of creates these little replication bubbles where it just happens in that one area all over the DNA. And this is where enzymes are going to become our best friends. So these are the key players in DNA replication. We have helicase, DNA polymerase, primase, and ligase. All four of these are enzymes. They're going to be very, very important in this DNA replication process. Okay, so step one. If I were you, it helped me when learning DNA replication to like kind of write out the steps and draw little pictures just to like help me understand. So that might help you guys. So basically what has to happen is we have to make the DNA code, the genetic code available to actually be replicated. So to do that, we have to unwind the DNA. So to unwind the DNA, we have helicase enzyme, which comes in. Enzyme comes in and basically unwinds part of the DNA helix. So it stabilizes is it by a single strand binding protein. Okay, so this helicase enzyme comes in and separates the helix into two, so that way then we can start replicating. So it comes in at both ends to help begin the replication. Now it creates this replication fork, which is kind of where it starts separating for replication to start. So step two is we have to prime the template. We have this enzyme called primase that synthesizes a strand of temporary RNA, which is just about 10 base pairs in length on the parent template strand. So DNA polymerase can't start replication without a 3' end to add onto. So the RNA primers are those temporary solutions. Okay, so since we split the DNA from the strands, we split them in two, we still need to try and create that opposite end or that opposite anti-parallel strand. So the RNA kind of comes in and acts as that solution temporarily, so that way the 3' end can be added onto. So step three, we have to actually build our complementary DNA. So let's say that this is our template strand. We have to build the complementary DNA to it. Okay, so you build the daughter DNA strand by just adding the complementary bases that match. So for example, here we have G, so the complementary base would be C. A to T, G to C, T to A, C to G. So DNA polymerase 3 is the enzyme that comes in and adds those bases. So this is an example of DNA polymerase. It's going to be moving its way up, okay, so that way we can add our different bases. So the DNA polymerases, DNA polymerase 3 adds on 1,000 bases per second, whereas DNA polymerase 1 adds about 20 bases per second. So DNA polymerase 3 is the main DNA builder because, 1,000 bases per second, very, very efficient. DNA polymerase 1 is pretty much the editing, repair, and then the primer removal. Okay, so that's the polymerase that's going to come in and remove the RNA primer and edit and repair any mistakes possibly made by DNA polymerase 3. So polymerase 1 was found by Arthur Kornberg in 1959, and polymerase 3 was found by Roger Kornberg in 2006. So father and son. So step 4, we're going to acquire energy to make bonds. So where does energy for bonding usually come from? ATP, okay? So what happens is we have to have a modified nucleotide. We break off one of those phosphates, and that's how we get the energy, ATP to ADP. Okay? But in because remember, ATP is considered a modified nucleotide, but we only have one phosphate group here in our base. Okay, so it's actually AMP. So the monophosphate, and we get the energy from breaking off those two. Now here, instead of ATP, we have GTP because it is the base of G-guanine. Okay, so the actual energy form would be GMP. T would be TTP, TMP, and C would be CTP, CMP. We get the picture, okay? So the energy of replication. 3 nucleotides arrive as nucleosides, okay? So it's DNA bases with 3 phosphate groups. There's 3 phosphate groups to have energy for the bonding. So they arrive with their own energy source for bonding, which is super convenient. Then they are bonded by the enzyme DNA polymerase 3. Okay, so they arrive in this fashion, all right? And then when you actually add the bases on, that's when to actually add them and bond them together, we break apart that phosphate or those other 2 phosphates, so that way we can get our appropriate nucleotide. So adding the bases, you can only add nucleotides to the 3' end of a growing DNA strand. So you need a starter nucleotide to bond. The strand only grows 5' to 3'. Okay, so the new strand will be 5' to 3' because you can only add to the 3' end. Okay, so this is how they come in. DNA polymerase chomps off those other 2 phosphates for energy, okay? And we just keep growing and building. Now we have leading and lagging strands, okay? So here the original enzyme helicase is coming in and growing in the replication fork. So it's basically still unwinding and opening up the DNA to continue to replicate. DNA polymerase is coming in. We have a 3' end here, so look, now we're going to start adding in 5' to 3'. Okay, boom, here. We have this complementary strand of DNA. But if you look over here, we cannot add to a 5' end. We can only add to a 3' end. So this is wrong and does not happen. So what's going to end up happening is we have these things called Okazaki fragments. That's named after the scientist who found out what they were, Okazaki. So this is our leading strand because that is the main one that is replicated correctly. This is going to be continuous synthesis. These over here, the Okazaki fragments, are called the lagging strand. So the lagging strand Okazaki fragments are joined together by ligase. It's kind of like a spot welder enzyme. What that means is it kind of just takes those pieces and glues them together as it goes along. So here is where we can get kind of like the replication fork or replication bubble that ends up happening. So this is the process of it all coming through and creating that bubble because it widens out. Here you can see the leading and then the lagging strand. Okay, here we go. Boom. So starting DNA synthesis, you have your RNA primers coming in. Okay, the RNA primers are coming in to actually help start out this process because you can only build onto a 3' end of an existing DNA strand. So you have primase RNA coming in to help start that all out. RNA primers are built by primase, which is an enzyme, and it serves as a starter sequence for DNA polymerase III. Primase is what helps DNA polymerase III add on those nucleotides. Step 5, we're going to replace the primers with DNA themselves. So DNA polymerase I is what's going to come in, remove those primers, and put the correct DNA nucleotides. But once again, it can only build onto a 3' end of an existing DNA strand. Okay? So you have loss of bases at the 5' end in every replication because the chromosomes get shorter with each replication. So there's a limit to the number of cell divisions. Now we have these things called telomeres. Those are repeating non-coding sequences at the end of chromosomes. It's kind of like a protective cap, like you would put a cap on a bottle. Okay, so it's a limit to about 50 cell divisions. That's called the Hayflick limit. So in DNA getting replicated, it's limited to about 50 phases of the cell cycle because you have that cap. So it's telomerase is an enzyme that extends the telomeres. Okay, so it can add DNA bases at the 5' end, but it has different levels of activity in different cells. So you have a high amount in stem cells and cancers. Stem cells, why? Because those are cells that haven't been differentiated yet. So why not freaking, like make them go crazy? And same with cancer. So these are kind of like the little caps that telomeres look like. So to edit and proofread DNA, 1,000 base pairs per second, that's a lot of typos. Okay, so we can have a lot of mistakes there. So that's where DNA polymerase I has a dual role, and it comes in and kind of does that repair and proofreads. It can remove mutated or damaged bases, and it repairs the damage consistently throughout life. And that actually reduces the error rate from 1 in 10,000 to 1 in 100 million bases. So thank God for DNA polymerase I. Okay, so we're pretty much done. So go ahead, pause, go through, answer these questions. Okazaki fragments. It's going to be A, DNA ligase. Pause, answer the question. Nucleotides during DNA replication is DNA polymerase. Okay. When DNA replicates, you go from a 3' to 5'. So which one is correct? It's going to be D. Remember the central dogma. This is how we go through the process, always. Okay, so it takes E. coli less than an hour to copy 5 million base pairs in its single chromosome. Divide to form 2 identical daughter cells versus human cells copying its 6 billion bases and divide into daughter cells only in a few hours. Our process of doing this is incredibly accurate, and it's only about 1 error per 100 million bases, so about 30 errors per cell cycle, which is freaking amazing that our bodies can do that. This is kind of how it happens with prokaryotes. Okay, obviously it's not going to be the same because they can, divide on their own through binary fission. So just kind of read through this on your own. Now we have epigenetics. Epigenetic factors are compounds that attach to or mark DNA. So basically they kind of interact with the genetic material, but just mark it. They act as chemical tags to say who, what, where, when, whatever genes should be turned on or actually expressed. And that's it for our DNA. Okay, next we're going to go over transcription and translation.